Okay, welcome back to chapter 20, number 2, screencast number 2. Dos más, ay, caramba. All right, okay, let's do it. Let's do it. Let's do it to it. Whoop, whoop. Whoop, whoop. Okay. All right. So last time we talked about how you clone genes, how you create recombinant DNA, transform that into bacteria, select for the correct bacteria using antibiotics, grow up that bacteria, and then you can study it in a lab. And you can make lots of cool animals, which we'll talk about in class. All right, so we also have 3 really important DNA analysis methods, which were mentioned at the very beginning, but we haven't really talked about them yet. All right, so the first one is called polymerase chain reaction. This is just a way of making millions of copies of a specific target DNA in the lab. And we can do this in about 2.5 hours or something like that. So it's not super slow. I mean, it's pretty quick. Make millions of copies, 2 hours. Sweet. All right. Second one is gel electrophoresis. This is a technique used to separate DNA based on its length, like how many base pairs it has. And then DNA sequencing is just determining the sequence of nucleotides. So what is the order of A, T, C's, and G's? And we're not really going to talk about the methods on how we do DNA sequencing, but it's really important to know that we now have the technology to sequence pieces of DNA. All right, so polymerase chain reaction, remind me in class. This is super important. I have to show you the song, not going to do it on the recording. All right, so there's a good video here that shows the basics. All right, and so what we're going to see is that in a polymerase chain reaction called PCR, a DNA molecule with whatever target sequence you want is going to be heated. All right, and what that is going to do is going to cause the 2 pieces of DNA to come apart. They're going to, it's called denature, right? So remember, protein denaturation is like losing its shape. DNA denaturation is going from double-stranded and they kind of separate, right? The second thing is that the mixture then cools. So we're going to see a lot of temperature changes. So we heated it up and now we're cooling it down. And so what the cooling is going to do is allow a DNA primer to bind. And all a DNA primer is a small piece of DNA which is going to allow for new replication. Okay, third thing here is that we are going to add our DNA polymerase, right? And so DNA polymerase, we know, is going to bind that primer and make new DNA, right? Remember, the primer is going to provide the 3' OH that is necessary for the DNA polymerase to move. All right, so we heat, cool, and then what we're going to see is that we're going to heat again. And this process is then repeated over and over and over again, usually, I don't know, 35 times or something like that. And by repeating this process many times, we're going to see millions of copies of DNA being made, right? So the 3 steps. So again, this was invented by a guy named Carey Mullis. And there's some crazy stories about he was on a motorcycle in the desert, potentially on LSD, riding around when he came up with this brilliant idea. He's kind of a wackadoodle. Anyway, so 3 repeating steps for PCR. Yes, you have to know these steps. Yes, you have to know what they do. 1, denaturation. So this is the first heat. So it's very hot, almost 100 degrees. So denaturation, right? That's going to separate the 2 strands from one another. You then anneal, which means we are going to add primers. Instead of saying add primers, I'm going to say primers bind, right? Remember, what does the primer do? It is going to supply the 3' OHs necessary for the next step, right? Third step is called extension. And this is where we are going to have a DNA polymerase actually create the new copy of DNA on both strands, right? So after one cycle, we have gone from one piece of DNA to two. What about after the second cycle? We're going to go to 2 to 4. What after the next? 4 to 16. 16 to, yeah, you get it, right? They're going to double each time. Oh, not 16, 8, blah, blah, blah. Yeah, they're going to double, okay? All right. I was going to say something important. Oh, important here. This is a heat stable DNA, right? Notice we're doing this at 72 degrees. So if we use the DNA polymerase in our bodies, which is about, what is it, 37 degrees, it wouldn't work, right? So these were actually isolated from hot springs. And this makes it possible, right? All right, so PCR is used in forensic science a lot. So let's say you had a single hair left at a crime scene. You are going to be looking for some sort of marker to determine if the criminal, if this piece of DNA matches any one of your three suspects, right? So this DNA can either be 200 base pairs, or it can be 300 base pairs, depending on the person, right? So what we're going to do is PCR and make many copies. So from a single hair or a single drop of blood, we can make billions or millions of copies of that DNA, right? So we are now looking at, I know this is the first time you have looked at a DNA gel. This is a DNA electrophoresis gel. But we're looking which suspect's DNA matches the DNA from the crime scene, right? So let's say here's the DNA from the crime scene here, right? And we're just looking which suspect matches that. And it looks like suspect 3 has the same piece of DNA, right? So we took DNA from 3 different suspects, and suspect 3 seemed to match the DNA from the crime scene. So it would be suspect 3, right? Question, why does suspect 2 have 2 bands? 2 different pieces of DNA from the same PCR? That is because they are a heterozygote, right? So they have 2 different copies, whereas suspect 1 and suspect 3 must be homozygous for different alleles. All right, so electrophoresis. So in gel electrophoresis, this is a technique to separate DNA based on size. We use what is called an agarose gel, right? And it is kind of, again, it feels like a hard jello. Yeah, a hard jello. And if you zoom in, it looks like a kind of like a sponge. It's got lots of little pores in it, right? So what we're going to do is we're going to put this gel and we're going to lay it down flat in a little contraption that has a power source. And in that gel, we have little holes at the top called wells where we can inject the DNA. All right, so the steps for gel electrophoresis, we have the DNA samples that are going to be injected or loaded into the well. We then add an electrical current through the power source. And remember, DNA is negatively charged. So those DNA molecules are going to start moving towards the positive electrode, right? The big key here is that large DNA is slower, small DNA is faster, right? And I always think of me and my son. Well, actually, now he's kind of big. I'm going to use my dog, Kenai. Me and Kenai both running through a super dense forest, right? Who's going to run faster and why, right? Kenai's going to be way faster. 1, she runs faster. 2, she's small and she can like dart between all the trees. And I'm like, trees and brush and all that. right? So a DNA agarose gel is similar. It's got all this stuff in it. So the small pieces are going to be able to go through much, much faster, right? And then we can stain the gel with a dye that binds DNA to see it, right? So here's an example. Remember, when we do the DNA gel, it's flat, like on a surface, but often when we're looking at pictures, we're going to look at them kind of upright like this. Right? So here, DNA samples are loaded up here into the wells. And then we are going to add a power source, add electricity, and those DNA fragments are going to move down to the positive electrode. And they are going to be separated by size. So again, the largest ones didn't move very far. The smaller ones moved quite far. And just to show you what that actually looks like, so here is an image of a PCR reaction. So that means we have many, many, many copies of a specific size. And what that results in is a band on the gel, which I'll describe later. All right, so in gel electrophoresis, we have a couple really important things. One is the ladder, and this is something that students have a really hard time with. So a ladder is a standard reference, right? It contains many pieces of DNA that have been cut to be specific lengths, right? So here we can see our ladder has one, two, I don't know. I'm kind of trying to highlight all of the different bands in the ladder. Each of those has a known size, a length, a known length, right? A band is a well-defined line of DNA on a gel. And it's important to note that bands contain many numbers of DNA. You cannot see a single piece of DNA on a gel. So for example, this is a band from a PCR reaction. And remember, this has millions of copies. And notice, it's very bright on the gel, right? That band is very noticeable. Whereas the ladder, these are not as bright because there's not millions of copies. There's a few less copies, right? So you then use your ladder to predict or to identify what size your PCR reaction is. So the ladder is used to determine your DNA size, all right? So what is the approximate size of our PCR reaction here? So I'm gonna look at my PCR reaction. Remember, it was loaded up here, so was the ladder. We added power, they ran down. When we stopped the power, the PCR reaction had gone this far. We are then going to compare that to the ladder, and it looks like it is a little bit bigger than 500, right? So if 500 base pairs is this lane or this band, I'm gonna say it's a little bit bigger. So I don't know, 600 or something, 550. All right, so let's check our understanding. All right, so which lane matches each description below? All right, so first one, this lane contains the longest DNA fragment. And we're looking at lanes 1, 2, 3, and 4. Remember, this lane here is our ladder of known sizes so that we can compare. So which one has the longest? That would be number 1, because remember, long equals slow. So it didn't go very far. So this one here is the biggest piece of DNA. And you can see that by looking at the ladder as well. This has over 3,000 base pairs. Second one, this lane contains the shortest DNA fragment. So again, we are going to look for the DNA that has traveled the farthest. That would be number 2. Remember, short equals fast. So it traveled the farthest. And then the third one, this lane contains a 1500 base pair piece of DNA. So we're going to look at the ladder. So we're going to look for which lane has a piece here. So we're going to move that over. And it looks like lane number 3 had that length. All right. So I just want to point out really quickly that these techniques are really important in forensics. All right. So often you can use mitochondrial DNA to kind of study a lot of that can help determine. Sorry, you can use mitochondrial DNA, not in addition to just your genomic DNA, right? And importantly, the mitochondrial DNA is only from the mother, whereas your nuclear DNA is from both parents, right? You can look at different lengths. So all of us have many repeats in our DNA, but people have different lengths of those repeats. So we can look at that. We can also do what's called DNA fingerprinting, which shows variations in these lengths of pieces cut by a given restriction enzyme. So for example, here we have a victim. It was a rape victim, all right? And we have a suspect, and we also have sperm found at the scene of the crime, all right? And we had victim cells at the scene of the crime. You can see that the two yellow here, we have the victim matches the victim cells, so that's good, that's a nice control. And we can now see that through DNA fingerprinting, each of the bands on the DNA from the sperm at the scene of the crime matched the suspect. So that shows that that is probably the perpetrator, right? GMOs, we're going to talk about in class. And for whole animal cloning, I just want to talk about this very briefly. Remind me to show you about this in class. It's amazing. Cloning your pet. All right, so people are always interested in Dolly when we talk about biotechnology. So what they did with Dolly is they took a cell donated from the udders of a sheep, and they grew that up in the lab, right? And what they did then from the other cell is that they took an egg cell donor and they removed the nucleus. So you took an egg and you removed the nucleus, and you took a cell of somebody you want to clone and you fuse that together. So now you're basically taking the nucleus of a cell you want and you're putting it into a donated egg that has no nucleus. You grow that up into an early embryo. You can then implant that into a surrogate mother, and you get a genetically identical cell to the donor, because that's the only source of DNA. And that's how Dolly was made. All right, and we will stop there. And remember, lots of practice questions. They're mostly from the Khan Academy. They're awesome. Use them. All right, bye.